Hello everyone, today's topic is environmental pollutants. Myself, Dr. A. Mazri, microbiology, government degree college, environmental pollutants. Nowadays, we can see there are different types of pollutants found in the environment, which are being a threat to many living organisms present in the environment, whether it is water, air, or soil. These pollutants are affecting humans, plants, and animals. As the population is increasing, industrial development is also increasing. As a result, the solid and liquid waste are being released into toxins in nature. These are non-degradable and accumulate in the environment. So many of the pollutants are degradable, but some of the pollutants are not degradable because there are no enzyme systems in the microorganisms, or there is no mechanism in the nature to degrade these pollutants, and as a result these for example plastic, as a result these pollutants are getting accumulated in the environment. Now after accumulation through food chain, the accumulated toxic substances are reaching in humans and causing variety of metabolic disorders. So when these are not degraded, they enter into the food chain through different organisms, and they are causing disorders, metabolic disorders in humans. Coming to the types of the pollutants, there are different types, that is complex organic compounds, inorganic compounds, natural products. What are the reasons for the accumulation? They are maybe dilution or diffusion or improper treatments. Like first coming to the dilution part, many of the factories or industries, what they do is whatever waste is coming from their industry, they will take this waste and add it to the water body, or they will release that waste into the water body. They think that dilution is the solution for disposal of the waste. Once it will be removed from the particular site, but it will be added to another site, that is water body. Next is disposal. Disposal means the waste from the place of generation is removed and it will be thrown at some other place. For example, these waste from households, they will be taken from the households and they will be thrown distant places. And the last one is improper treatment. So many of the waste, for example, plastic waste, they will burn the waste. So this is not the proper treatment of the waste. In the same way, there are many improper treatment methods by which these pollutants are causing present use. In spite of environmental pollution due to anthropogenic activities, anthropogenic means due to human activities. So what are the different types of pollutants? If we take here, three types of gases: nitrogen oxide, sulfur dioxide, carbon dioxide, and particle pollutants are placed in atmosphere and greenhouse gases. All these are entering into the air from different sources and then into the soil. You can see hazardous waste, oil waste, persistent organic pollutants, in soil activity due to massive saving. And if you take water, chemical and biological pollutants are released into the water in case in domestic waste types, oil waste, eutrophication, caused by nitrogen and phosphorus sources. So all these sources are responsible for putting waste from soil and water. What are the main factors in bioremediation process for this? Bioremediation means treating these pollutants. First environment should be amicable in nature, whether it's agriculture, also can be on temperature or the degradation process and contaminants toxicity consumption by the soil. They must be helpful they must be in nature in manufacturing that it helps breakdown and the microorganisms so they must be metabolically active and they must be capable of degrading the toxin and degrading population next to indigenous population organisms which are taking that outside. If they are able to degrade that particular waste, it will be more helpful because they will increase transportation hydrocarbon, shifting the waste from one place to another place. And the genetically modified microorganisms, if we can use them, it is more helpful. And we can have different genes for the using different types of the compounds. Next, we have to understand what is xenobiotics. Xeno means foreign, broadly referred to the unnatural, foreign, and synthetic chemicals such as pesticides, herbicides, fertilizers, will find them other compounds. If you want to understand the word xenobiotic. Xeno means a foreign. It broadly refers to the unnatural, foreign, and the synthetic chemicals. Means they are not present in the nature from the starting, and they are foreign to the nature, and they are synthetic chemicals which are made in the labs. For example, pesticides, herbicides, pesticides, solvents, and other organic compounds. The microbial degradation of the xenobiotic compounds assumes significance since it provides an effective and economic means of disposing of toxic chemicals, particularly the environmental pollutants. So, when these xenobiotic compounds are degraded by microorganisms, provides an effective and economic means of disposing of toxic chemicals. Means financially it will be very feasible and effective because there won't be any side effects or side products due to this degradation process. And Pseudomonas are predominant microorganisms for bioremediation. Out of many microorganisms, scientists have found that Pseudomonas is the microorganism which is responsible for degradation of many compounds in nature. Members of the genus Pseudomonas are the most predominant microorganism that degrades xenobiotically. It is found that this Pseudomonas is able to degrade more than 100 organic compounds. Different strains of Pseudomonas are able to detoxify more than 100 organic compounds, and the examples of organic compounds which are being degraded by Pseudomonas are several hydrocarbons, phenols, organophosphates, polyfluorinated biphenyls, PCBs, and polycyclic aromatics, and methanol. So all these compounds are being degraded by Pseudomonas. It is an important microorganism. And there are other, apart from Pseudomonas, there are other microorganisms which can degrade these xenobiotics. For example, so we have studied about Pseudomonas. Coming to the Mycobacterium species, it is able to degrade the compounds such as benzene, branched hydrocarbons, paraffins, etc. And intelligent species are able to degrade polyfluorinated biphenyls, alkyl benzene, halogenated hydrocarbons, etc. Nocardia species is able to degrade naphthalene, alkyl benzene, monoxyacetate, etc. Like this is the list of the microorganisms, and these are the list of the chemicals which can be degraded by these microorganisms. Next, consortia of microorganisms for biodegradation. Consortia of microorganisms. Single strain of microorganism may degrade one or more compounds. Like if we take one microorganism, it may degrade one microorganism, um, one compound. cases, for degradation of a particular compound, many microorganisms are required. Sometimes for the degradation of single compound, the synergistic action of a few microorganisms, that is a consortium or a cocktail of microbes, may be more efficient. Okay, so for the degradation of a particular compound, many microorganisms cocktail is sometimes required. Example, the insecticide parathion is more efficiently degraded with a combined action of Pseudomonas aeruginosa and Pseudomonas stalzeri. Okay, so parathion degradation requires combined action of both species, that is Pseudomonas aeruginosa and Pseudomonas stalzeri. It means no single organism out of these two is effective in degrading this insecticide parathion simply. Next, co-metabolism in biodegradation. Co means together, metabolism. Both break down the compound together. The metabolism of xenobiotics, no advantage to the microorganism. So when these microorganisms are breaking the xenobiotics, there is no benefit for the, no advantage for the microorganisms, as it cannot serve as a source of carbon or energy for the organism. When the xenobiotics are broken down, they are not serving as a source of carbon or energy for the organisms. The term co-metabolism is often used to indicate the non-beneficial biochemical pathways concerned with the biodegradation of the xenobiotics. Co-metabolism means it indicates the non-beneficial biochemical pathways which are involved in the degradation of xenobiotics. However, co-metabolism depends on the presence of a suitable substrate for the microorganism. Such compounds are referred to as co-substrates. Here we have understood that the microorganism which is breaking down the xenobiotic, that microorganism is not getting any advantage out of this breakdown process of xenobiotics, because these xenobiotics are not serving as a source of carbon or energy. But the term metabolism means non-beneficial biochemical pathways for degradation of xenobiotics. However, co-metabolism depends on the presence of suitable substrate along with these xenobiotic compounds. No doubt these microorganisms are breaking these xenobiotics, but there should be some substrates present in the vicinity of these xenobiotics, which can serve as a source of energy and carbon for these microorganisms. Such as substrates or such compounds are called as co-substrates. So now we have clear the concept of co-metabolism. Next, factors affecting biodegradation. Several factors influence biodegradation. Chemical, of which first one is chemical nature of the xenobiotic. What is the nature of the xenobiotic? Chemical nature, it is more important for the degradation process and the capability of the individual microorganism. Whether the particular microorganism is able to degrade the particular xenobiotic or not, and the nutrient and oxygen supply. Just as we have discussed, whether the microorganism is able to get any nutrients out of the xenobiotic and oxygen supply role. The degradation process is aerobic or anaerobic, and what temperature is required for the degradation process, and pH is also required, suitable pH is required, and redox potential also plays an important role in this degradation process. Next, some of the relevant features for this process are aliphatic compounds, easily degraded than aromatic ones. Aliphatic means straight chain compounds, and aromatic means cyclic ones. So when compared to cyclic compounds, straight chain compounds are easily degraded. Presence of cyclic ring structures and lengthy chains or branches decrease the efficiency of biodegradation. So when in xenobiotic compound, when the cyclic rings are present or branch points are present or when the chains are very lengthy in nature, it will be difficult for the microorganism to degrade such compounds, and water soluble compounds are easily degraded. Okay, so water soluble, if the compounds, if the xenobiotics are water soluble, then it will the degradation will become more easy for the microorganisms, and molecular orientation of aromatic compounds also plays an important role in this biodegradation. Molecular orientation, that is whether the compound is ortho, para or meta. We understand the arrangement of two groups on the benzene ring. If they are in at a particular position, they are called as ortho. Then para, then meta. This is the order of degradation of the compounds. And next is the presence of halogens in aromatic compounds inhibits biodegradation. Halogens are the seventh group compounds: chlorine, fluorine, bromine. If they are present in aromatic compounds, then the degradation will become more difficult. Now, let us revise these compounds. Aliphatic compounds are more easy than aromatic ones, and presence of ring structures or branches or lengthy chains is also difficult. Water soluble compounds are easily degraded. Molecular orientation plays an important role. Presence of halogens makes it difficult for the degradation. Next, you have to understand the term biostimulation. This is a process by which the microbial activity can be enhanced by increased supply of nutrients or by addition of certain stimulating agents. Example, electron acceptors and surfactants. Here, the process of degradation by the microorganism is increased by adding certain substances, or the process of biodegradation is stimulated or initiated by adding certain substances. Such substances are called as stimulants. Next, bioaugmentation. It is possible to increase biodegradation through manipulation of genes. Can also be achieved by employing a consortium of microorganisms. Bioaugmentation means again this is also a process to improve the biodegradation process through manipulation of genes. Means genes can be changed in the microorganism, or in some cases a consortium can also be employed. As if we can employ the consortium, that also consortium means group of microorganisms are involved in the breakdown process. That is called as bioaugmentation. Next is enzyme systems for biodegradation. So several enzyme systems with independent enzymes that work together are in existence in the microorganisms for the degradation of xenobiotics. As we have discussed, the enzymes are required for degradation of xenobiotic compounds. So where these enzymes are present, they are present in the microorganisms. Then the genes coding for the enzymes of biodegradative pathways may be present in the chromosomal DNA or more frequently on the plasmids. So whatever genes which are present, which are required for the particular enzymes, which help in biodegradation pathways, they may be present in the chromosomal DNA or more frequently on the plasmids. We know that if an enzyme has to be synthesized, there should be some gene responsible for the synthesis of that particular enzyme. So that responsible genes, they may be present on the main chromosomal DNA or a plasmid DNA. Next, in certain microorganisms, the genes of both chromosome and plasmid contribute for the enzymes of biodegradation. The microorganism Pseudomonas occupies a special place in biodegradation. So, in some microorganisms, genes may be present on the main chromosome or the plasmid DNA, or in some microorganisms, both the chromosomal genes as well as plasmid genes will help or will contribute for this degradation process. Next, a selected list of xenobiotics and the plasmid containing genes for biodegradation. Example, this we have taken Pseudomonas. So this is the xenobiotic compounds. Okay, naphthalene, xylene and toluene, salicylate, camphor, chlorobenzene. All these xenobiotics, they require certain genes. For example, for naphthalene, the gene which is present on the plasmid of Pseudomonas is NAH. For xylene, gene required is XYL. Xylene and toluene are degraded by TOL, WWO, XALK, and salicylate requires the gene FAL. Camphor requires the gene CAM. Chlorobenzene requires CAC2C25. So these are the genes which have to be present in the concerned microorganism, which they are present in the pseudomonas, and they are responsible for synthesis of enzymes for degradation of xenobiotic compounds of this variety. Next, recalcitrant xenobiotics and reasons. What do you mean by the word recalcitrance? Means the compound is not able to undergo degradation, or the compound is resistant for degradation. So why these xenobiotics? Xenobiotics we have understood these are the foreign compounds which are not present in the nature from the starting. They may be referenced manually in the lab by the humans. So these xenobiotics, what is the reason for the xenobiotics recalcitrant? Why these xenobiotics are not being what are the reasons? These are not easily biodegradable and therefore persist in the environment for a long period. They are labeled as recalcitrant. So basically, they are now degraded. So what are the reasons? They may be chemically and biologically inert. Inert means they are not reactive. Inert means they are not reactive. Even in periodic table, we see some elements are inert elements. Right. So these xenobiotics, they are stable, highly stable. That is why they are not able to get broken down by any enzymes or any microorganisms. Lack of enzyme system in the microorganisms for biodegradation. Another important point is the microorganisms may be lacking enzyme systems. Maybe they are not having any enzymes for degradation of these legal biotics. That is why they are not getting degraded. They cannot enter the microorganisms being larger molecules or lack of transport system. Sometimes the reason is they are not able to enter into these xenobiotics, enter into the microorganism or into the microorganisms because of their larger size, and there is no particular transport system for transport of these compounds from the outside of the cell to inside of the cell. The compounds may be highly toxic or result in the formation of highly toxic products that kill microorganisms. So sometimes these compounds may be highly toxic, or if they break down, they may release some toxic chemicals which are responsible for killing of microorganisms. So that may be the reason why the xenobiotics are not getting degraded. Next, recalcitrant xenobiotic compounds are chloroforms, freons, insecticides, that it is DDT, linden, herbicides, dalapone, example, and synthetic polymers such as plastics, which consists of polystyrene, polyethylene, polyvinyl chloride. So these are few examples of recalcitrant xenobiotic compounds. It takes about 4 to 5 years for the degradation of DDT, out of which 75 to 100% may be degraded in the soil. But for this degradation of DDT, a group of microorganisms such as Aspergillus flavus, mucor, alternans, Fusarium oxysporum, and Trichoderma viridie are responsible for degradation of DDT. Uh, Next term is biomagnification. Bio means life, magnification means increasing the content. Here, You can understand this term in such a way that here something is increasing in living organisms. Progressive increase in the concentration of xenobiotic compounds. We understand basically that xenobiotic compounds are not getting degraded, but as a result, when the concentration of the xenobiotic compounds increases gradually in the food chain, when it passes through the food chain, as the concentration of the xenobiotic increases, that process is called as biomagnification or bioaccumulation. Example is DDT. When they are eaten by fish and birds, this pesticide being recalcitrant accumulates and enters the food chain. Right. So normally they enter into fish and birds through water. So this pesticide being recalcitrant, as it cannot broken down, it accumulates and enter into the food chain. The DDT may find its entry into various animals, including man. DDT affects the nervous system and it has been banned in some countries. Even in journal, one of the journal, it was stated that DDT has also been found in mother's milk, which was considered to be one of the purest compound on this earth. And effect of DDT use in Borneo. In the early 1950s, the people in Borneo suffered from malaria. WHO had a solution: kill the mosquitoes with DDT. So this is what has happened. So when in Borneo, this DDT was used to kill the mosquitoes, then it has eliminated byproduct 1, eliminated the natural predator for the catch eating caterpillars. So caterpillars, these parasitic wasps are killed. As a result, caterpillars population has been increased. By the increase of caterpillars, these caterpillars ate the roofs of the villagers' houses, and as a result, the cats died. The rat population flourished. So did an outbreak of plague has resulted. Eating the geckos caused more DDT to accumulate in the cats that eventually that killed the wild cats. Then DDT accumulated in geckos. So this is the byproduct too. Caterpillars number has increased and cats have died, and geckos, due to accumulation of DDT in geckos, cats have died. So rats have increased and it has resulted in plague. Next, coming to the synthetic polymers or plastics. About 90% of the plastic materials, mainly disposable goods and packaging materials such as polyethylene, polyvinyl chloride, and polystyrene, that appears to resist biodegradation. So these compounds, they are not undergoing any biodegradation because of larger molecular size or excess molecular size. And if we can reduce this molecular size into short polymer chain fragments, for example, by pyrolysis fragments, the short fragments will become biodegradable. Okay, so for some of the plastic materials, if you can break down the compound into lower molecular weight fragments, then it may undergo degradation. Next, the second alternative to produce biodegradable plastics. So the plastic nature, the material used for the synthesis of be somewhat biodegradable in nature. For example, PHB, that is polyhydroxybutyrate, which is similar to synthetic polyesters used in textile industry. It is the actually PHB is the storage compound present in many of the bacteria. Example, alkaligens eutrophus. So this PHB has been used in the past for synthesis of surgical thread, which is used for giving stitches after operation for the patient after undergoing any operation for stitches. Okay, so the benefit of this PHP is this is naturally degraded within the skin. There is no necessity of the pain for the patient to go again to remove the test. So this is naturally dissolved in this body, human body. So that is biodegradable. And the bacterial PHB is rather fragile but is biodegradable, so makes disposal easier. So one of the reason for this bacterial PHB is it is somewhat fragile. So we have to find alternative means and sources to make a biodegradable plastic. This compound can be processed to form plastic products. It is used in surgical sutures. As I have discussed, it is used in surgical sutures instead of threads, which can later dissolve once their job is over. However, PHB is now being manufactured in tons and should soon become a large-scale commercial product to compete with plastics now used. So, we know very well what are the adverse effects due to plastic accumulation in the environment. So, we have to find an alternative, and PHB is one of the important alternative which can help in overcoming this plastic pollution. And it is a good news that it is being synthesized in larger quantities so that it may compete with the plastic. Next, applications of PHB or polyhydroxybutyrate. In the biomedical field, it is used for surgical thread, and it supports the tissue cultures for implants, surgical implants, dressings, parts of bones, and replanted veins, engineering of hardware, and things, many more things are there. Its pharmacological area, it is used in encapsulation for medicines and control helps in which helps in control release of the medicine in the human body. Next is packaging, means food packaging. Veterinary area, it is used for again encapsulation for veterinary medicinal products and agriculture. It is used for encapsulation of fertilizers. In the environment, it is used as bags, bottles, disposable items, and the items for personal hygiene, films of environment, degradable diapers, and remediation of areas affected by oil spills. Wherever oil spills are there, they have used it PHP. Next, industrial area, PHB has been used for recovery of oligomers and monomers for new use in synthesis of polymers. Next, coming to the oil spill. Oil spill, first we have to understand that this crude oil, whatever we are getting, petrol, diesel, whatever we are using, we are not generating it here. We have to transport that, or we have to import that product from other countries such as Saudi, etc. So during transport, this transport is mainly done through water bodies, that is sea. And during this transport, sometimes there can be accidents. So due to that accident, oil spills will be there. So whenever there is an accident and when oil spills on the water body, it will remain as a layer on the water body, which prevents oxygen to enter into the water body, and which is killing many of the organisms which are present in the water or aquatic environments in the depth of the sea. So extraction, production, transport, utilization, and disposal of fossil fuels. Oil is next. Oil pollutants undergo different physical and chemical changes during regenerative processes. So once if after the oil spills, if it try to undergo um, degradation also, it will undergo some different physical and chemical changes which are not good for the environment. And oil fuels include complex mixture of aliphatic, alicyclic, and aromatic hydrocarbons and some non-hydrocarbon compounds such as naphthalenic acid, phenols, thiols, and heterocyclic nitrogen. So what these oil spills or oils contain? They are containing aliphatic compounds, alicyclic compounds, or aromatic compounds. They can contain straight chain compounds, cyclic compounds, or benzene compounds and some non-hydrocarbon compounds also they are having. Next, degradation of these compounds is really a challenging task for microorganisms because the list of compounds which are present in these oils are many number. And so if there is a single compound in this oil, we can think of degrading that particular compound. But as we have seen, there are many number of compounds present in this oil. So for degradation of a number of compounds, we should have a number of tools or a number of enzymes for the degradation. For that matter, we should also have a number of microorganisms which can degrade all the compounds. So now crude oil sample is subject to complete biodegradation. Whatever till now we have seen that any crude oil there was no 100% degradation in any area. Extensive degradation of petroleum mixture requires the combined activities of several different microbial populations. As I have told you, as there are a number of compounds, many number of compounds are there, many number of microorganisms are required. Their efforts are required for the degradation of this oil spill. And important biochemical reaction during degradation of hydrocarbons is the secretion of mono and dioxygenases to clean the benzene ring. So what is important here for the as I have told you, hydrocarbons are there. So for the breaking of these hydrocarbons, some enzymes are required. These enzymes are called as monooxygenases and dioxygenases. Monooxygenases and dioxygenases. So these enzymes they will cleave the benzene ring. Okay, they will cleave monooxygenases and dioxygenases enzymes can cleave the benzene ring. These enzymes catalyze the incorporation of molecular oxygen into the benzene ring which results in the ring cleavage. So what these mono and dioxygenase are doing, they are making the oxygen or adding oxygen to the benzene ring so that the ring can be cleaved. The dioxygenase is catalyzed by the incorporation of two atoms of oxygen into the benzene nucleus, while monooxygenase is catalyzed by incorporation of one atom of oxygen. So the names of the enzymes monooxygenase or dioxygenase, what they are doing. Monooxygenase is inserting one oxygen atom into the benzene ring, whereas dioxygenase is inserting two oxygen atoms into the benzene ring. That way they are degrading the benzene ring. Next, here Pseudomonas mendocina attacks the aromatic hydrocarbon toluene by different mechanisms. Here we can see this is the benzene ring or toluene. Methyl group attached to the benzene ring is called as toluene. So here we can see to this ring two places. At two places OH group or hydroxyl is oxygen is added, hydroxyl group is added. So this enzyme involved here is a dioxygenase. Okay, this is the dioxygenase. And one more method is here oxygen is added here at this position. So by this addition, the ring cannot be cleaved. And the another position, another mechanism is the hydroxyl group is added at the base. So it is one hydroxyl group is added to the ring. So this is done by monooxygenase. This is done by monooxygenase. Here it is done by dioxygenase, right? Direct dioxygenase attacks on the ring leads to 6-dihydrodiols formation. This one. Next, methyl group oxidation leads to benzoic acid. This when this methyl group is oxidized, it will lead to formation of benzoic acid formation and monohydroxylation of the ring gives para-result. Next one is laundry detergents. In many detergents, many chemicals are being used in laundry detergents. One of the chemical used in laundry detergent is alkyl benzyl sulfonate. Alkyl benzyl sulfonate. So this component is a major component used in laundry detergent. And here, as the name suggests, it consists of alkyl group, benzyl group, and sulfonate. So alkyl portion of this ABS, in short, we can call this alkyl benzyl sulfonate as ABS. Alkyl portion of ABS molecule is branched, that is non-linear, and proves to be recalcitrant and causes extensive foaming in water bodies. So when we wash, when we use the detergents, our clothes, these chemicals which are used in detergents are also released into the water bodies, right? Which are causing extensive foaming in the water bodies and leading to water pollution. So this foaming is mainly due to alkyl portion of the ABS molecule, and this alkyl portion of ABS molecule is a branched one or non-linear one. Scientists have modified this alkyl portion of ABS from non-linear or branched form to linear form. Non-linear form to linear form. The design has been changed from non-linear to linear form. Then the alkyl benzene sulfonate turns to be more easily biodegradable. Okay, so in this we can understand a part of the compound, or a part of the compound it has been changed, then the entire compound is getting degradable easily, biodegradable easily. Detergent industries of many advanced countries have switched from non-linear to linear ABS to overcome the problem. So many advanced countries have gone for linear structure of ABS to overcome the pollution caused by alkyl benzene sulfonate. Next important component is biodegradation of the pesticides. So we know very well what are pesticides. As the name we can, if you break this word pest, pest means the insecticides or uh, which damage the crops. Pest side means killing. The chemical which can kill the pests are called as pesticides, right? So where ill effects of these pesticides are disturbance in biological equilibrium. When we are using these pesticides, biological equilibrium is getting disturbed, and there is an increase of disease susceptibility, and there is also bioaccumulation. Means these chemicals are getting accumulated in the living food chain. Development of tolerance, and people are getting tolerance. If you want to remove this compound, the tolerance levels are increasing. Disturbance in reproductive physiology. Many infertility problems are due to these pesticides. Effects on behavior. Mental behavior is also changing. Effects on wildlife, fish, and other aquatic organisms. And plants are also being affected. Now, these pesticides are applied on crop fields generally. Only 0.1% of the substance is sufficient to kill the target organisms, but remaining 99.9% is spread over in the environment. Whatever amount of the pesticide they are spraying in the crops, out of which only 0.1% is helpful in killing the pest, whereas 99.9% is causing damage to the environment through the food chain. It ultimately reaches the humans. So what happens from the crop? It will gradually enter into the water body. From there, it will enter into the food chain. Many pesticides are non-biodegradable or sparsely degradable. So these pesticides are also mostly non-biodegradable. They remain in the environment for longer time in toxic form, inflicting the adverse effect on the ecosystem. So they will remain in the environment for many years, resulting in damage for the ecosystem, and bioaccumulation or biomagnification can also be seen due to pesticides. So these are the different pesticides, and these are the persistence times. If you can see DDT, it is being in the nature for more than 15 years. This is the most popular pesticide, and aldrin and all these are endrin. All these remain in the year for more than 14 to 15 years, and as it is for different another pesticides, they have given the time, persistence time in the environment. So how these pesticides can be degraded by using microorganisms. So Pollack, the scientist in 1974, has suggested four main possibilities for transformation or inactivation of these pesticides by microorganism. The pesticides is used as a substance and energy. So one possibility is there must be some microorganism which can use these pesticides as a source of energy or food. The pesticide can undergo co-metabolism, that is, organism transform it but cannot derive energy for growth from it. And we have seen co-metabolism. If we can supply pesticide along with the other substance from which the microorganism can derive energy or carbon source, this can also be broken. Next, the entire pesticide molecule or its intermediates can be conjugated with naturally occurring compounds. One of the possibilities is we can club this pesticide with a naturally occurring compound. The pesticide is incorporated and accumulated within the organism. Next, so these are the major classes of pesticides, and these are the pesticides compounds. Next, there are two types of processes. One is enzymatic and non-enzymatic, and the other one is non-enzymatic process for microbial metabolism of pesticides. Incidental metabolism, which serves as an energy source. Sometimes pesticides are serving as a source of energy. Some enzymes are also involved in this degradation of pesticides, and we can see in nature co-metabolism is operating, and catabolism. Insecticide serves as an energy source, and detoxification is also there in the nature. The non-enzymatic patterns are divided into photosynthetic breakdown, contribution via pH change, production of organic and inorganic reactants, production of cofactors. All these conditions will help in non-enzymatic breakdown. They are also These conditions are also helping breakdown of the pesticides, but they are non-enzymatic in nature. Then next is microbial enzymes required for degradation of the pesticides. These are the bacteria which can degrade all these pesticides, and these are the fungi which can degrade these pesticides. So these microbes are having certain systems for the degradation of particular pesticides. And biochemistry of pesticidal degradation. Here we can see aliphatic carbon chains, means straight chain carbon chains are degraded by beta oxidation sequence, and the resulting C2 fragments are further metabolized through PCA cycle. There is a step-by-step process of breaking down of any pesticide. If it is an aliphatic carbon chain, means straight chain carbon chain, lengthy chain is there. First, it has to be broken down at the second position. Beta means alpha means first position, beta means second position. At the second carbon position, it must be broken down. As a result of two carbon fragments or C2 fragments will be released. These C2 fragments are further metabolized or utilized through PCA cycle. Next, the substituent of aromatic rings is removed partially or completely. When we want to degrade aromatic ring compounds or benzene ring compounds, first we have to deal with the substituent groups. First, they have to be partially removed or completely removed, and then aromatic ring structures must be degraded by dihydroxylation and ring cleavage mechanism. We have seen the enzymes responsible for breaking of the enzyme ring, that is monooxygenases and dioxygenases, which can be operative here. If substituents and organic compounds are halogens, nitro or sulfonate groups, then they are unnatural, so they cause interface in oxygenation and cause recalcitrance. Sometimes if the groups which are present in the benzene ring or halogens or nitrate groups or sulfonate groups, they also create some problems, and they make the compound recalcitrant or difficult for degradation. If substituents are methyl, methoxy, carboxyl or other groups, they can be removed from blocking positions metabolically. So they can be removed by blocking their positions metabolically. Next, as time is increased, so we'll continue in the next session. Thank you for your patience.